Hello everyone, this is our third tutorial on sequence alignment. In the previous tutorials, we have aligned the reads to the reference genome and also marked the duplicates, and also we looked into the statistics of the alignment. In this tutorial, we will explore this alignment file through visualization. For that, we will be using Integrative Genomic Viewer IGV. This is a high performance visualization tool that is developed to visualize different types of genomic data. This visualization helps us to understand and look into the details of the genomic data. Genomic data is complex and it is stored in a much bigger file. Such visualization will help us to understand and to comprehend the details of the genomic files and the data. IGV tool can be used to visualize different kinds of genomic data, such as to look into the alignment and to look into the tracks of epigenomic data and to visualize the RNA-seq data, copy number alteration from whole genome sequence data. Also, we can visualize variants and genotype information. To visualize diverse genomic datasets, IGV accepts different kinds of files, such as it accepts BEM file, BED file, SEM file, BED graph file, FASTA file. It accepts many different files so that we can explore those data within this file using IGV. IGV is an open source software. It is freely available and it has different versions such as desktop application and web browser. Here is a screenshot of the web browser. We can upload our data onto this web browser. Today we will work on the desktop application. We will install the IGV desktop software into our computer and we will upload the data that we have generated in our previous tutorial. Installing IGV is very straightforward. For that, we need to go to this download page of IGV, and my computer is a Windows computer, so I will download this IGV for Windows, Java included. Let's download this executable and install it into the computer. This is the installer file. We can easily install by double clicking. Accept. IGV has been successfully installed. So this has created an icon onto my desktop. I will open this. This is the interface of IGV. IGV allows to upload files. We can upload file by clicking the button, and also we can save session. We can export the visualization using PNG or SVG file format. Within IGV, we need to upload the genome. We can upload any genome. There are already some genomes hosted. For today's tutorial, we will look into the data of SARS-CoV-2. So I will link the SARS-CoV-2 genome. Click OK. And there are some options to change the view preferences and the layout of IGV. And if we have multiple tracks, we can sort them, we can rename them. We can also navigate the specific genomic regions. There are other tools that are integrated within IGV. For example, we can use BLAT to search small DNA sequence matches with the reference genome, and there are help options. Also, there are other buttons to select, to highlight, and this button is very helpful to zoom in or zoom out. Now let's upload the BEM file that we have aligned in our previous tutorial. I'll select the BEM file. The BEM file is uploading. Yes, this is how it looks when we upload a BEM file to IGV. Here we are showing the entire coronavirus genome. The bottom panel is showing the genes that are present in the coronavirus and their location across the genomes. The top panel here is showing the number of reads that are mapped to this particular location. On average, there are more than 3000 reads per genomic location for this library. And here, all the reads are represented in gray bars. We can see there are thousands of reads that are mapped to the genome that are showing in gray bars. We can scroll down. Each gray bar here is an individual read. If we click on one of the reads, it will show the detailed information about the read and the alignment. Let's go to the top. In this visualization, gray color is indicating that those reads or the sections of the reads mapped perfectly to the reference genome. We can see some discrepancies in color. Those discrepancies are indicating the differences in those reads compared to the reference genome. Let's take a look into this red line. There is a red line in most of the reads. We will zoom in to investigate further. We can drag, we can zoom in. There is a T. There is a T base in most of the reads where it is G in the reference. So this difference has been visualized using a different color of base. So if a read is perfectly matched, those are in gray color. Let's take a look into this. There is an I sign which is indicating an insertion of a base in this particular location for this read. Let's take a look into an example of deletion. There is a nucleotide that is deleted in this read. So a deletion is represented with a black line. I want to show another feature which is changing the color of reads by different conditions. Here you can see some reads are red. So because we have colored by insert size, if the insert size is outside of the expected range, it will show in a different color. We can also change color for other conditions such as for read group information, which is same because all the reads have same read group information. I would recommend you to look into other conditions and to change the color of the reads to explore further this library. Finally, I will show one thing. For example, if you want to save this file, this visualization, we can actually save it. This will save this entire window. Today, I will not show other functionalities of IGV because we wanted to show the visualization of alignment. In our future tutorials, when we will work with ChIP-seq, RNA-seq, or whole genome sequence data, we will show different functionalities of IGV, and we will be using IGV to visualize our complex data. Thank you for watching this video. See you in the next one.